जय श्री राम दोस्तों। तो चैप्टर का नाम है सेल द यूनिट ऑफ लाइफ। इंट्रोडक्शन से स्टार्ट करते हैं हम। सबसे पहले इंट्रोडक्शन। व्हेन यू लुक अराउंड यू सी बोथ लिविंग एंड नॉन लिविंग थिंग्स. You must have wondered and asked yourself what is it that makes an organism living, or what is it that an inanimate thing does not have, which a living thing has. The answer to this is the presence of basic unit of life, the cell in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others like us composed of many cells and are called multicellular organisms. What is cell? Unicellular organisms are capable of independent existence and performing the essential function of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, a cell is the fundamental structure of and a fundamental unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural detail of the cell. Cell theory. In 1838, Matthias Kleiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells, which form the tissue of the plant. At about the same time, Theodore Scaven, 1839, a British zoologist studied different types of animal cells and reported that the cells had a outer layer which is today known as the plasma membrane. He also concluded, based on his study on plant tissue, that the presence of cell wall is a unique character of the plant cell. On the basis of this, Scaven proposed the hypothesis that the bodies of animals and plants are composed of cells and product of cells. Scladen and Swan together formulate the cell theory. This theory, however, did not explain as to how new cells were formed. Dolph Virchow 1855 first explained that cells divided and new cells are formed from pre-existing cells, omnis cellula e cellula. He modified the hypothesis of Scladen and Swan to give the cell theory a final shape. Cell theory is understood today is all living organisms are composed of cells and product of cells. All cells arise from pre-existing cells. An overview of cell. You have earlier observed cells in an onion peel and or a human cheek under the microscope. Let us recall their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human chick have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains the chromosomes, which in turn contain the genetic material, DNA. Cells that have membrane bound nuclei are called eukaryotic, whereas cells that lack a membrane bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. Cytoplasm is the main area of cellular activity in both the plants and animal cells. Various chemicals reaction occur in it to keep the cell in the living state. Beside the nucleus, eukaryotic cells have other membrane-bound distinct structure called organelles, like the endoplasmic reticulum, the Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both prokaryotic and eukaryotic as well. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles chloroplast in plants and mitochondria and on rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelle called centrosome which help in cell division. Cells differ greatly in size, shape and activities. For example, mycoplasmas, the smallest cells, are only 0.3 micrometer in length while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be dish-like, polygonal, columnar, or cuboidal, thread-like, or even irregular. The shape of the cell may vary within the function they perform. Prokaryotic cells. Prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, PPLO, pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, rod-like, coccus, spiral, uh, vibro, coma-shaped, spiralum, spiral. The organization of the prokaryotic cells is fundamentally similar, even though prokaryotic exhibit a wide variety of shapes and functions. All prokaryotics have a cell wall surrounding the cell membrane except in mycoplasma. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not involved by a nuclear membrane. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have small circular DNA outside the genomic DNA. The small DNA is called plasmids. The plasmid DNA confers certain unique phenotype characters to such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotic. No organelle like the ones in eukaryotic are found in prokaryotic cells except for ribosome. Prokaryotics have something unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is a characteristic of prokaryotes. They are essentially enfolding of cell membrane, cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure. That is the outermost glycolex followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the difference in the cell envelopes and the manner in which they respond to the staining procedure developed by Graham-Witch. Those that take up the gram-stain are gram-positive, and others that do not are called gram-negative bacteria. Glycolex differs in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in others it may be thick and tough called capsule. The cell wall determines the shape of the cell and provide a strong structural support to prevent the bacterium from bursting and collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of eukaryotes. A specialized membranous structure is the mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. They help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration secretion process to increase the surface area of plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: a filament hook basal body. The filament is the longest portion and extends from the cell surface to the outside. Beside flagella, pili and fibrae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structure made up of special protein. The fibrae are small bristle-like fibers protruding out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also the host immune. Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits 50s and 30s units, which then present together form 70s prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes and polysome. Polyribosome और polysome में से कुछ भी बोल सकते हैं. The polysomes of a polysome translate the mRNA into protein. Inclusion bodies. Reserve materials in the prokaryotes are stored in the cytoplasm in the form of inclusion bodies. They are not bounded by any membrane system and lie freely in the cytoplasm. Example: phosphate granules, cyanophysian granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. Eukaryotes.